Dr. Kirsch, who is the inventor of the Alpha-Stim CS and who introduced Alpha-Stim to the market, will now present data evidencing the safety and effectiveness of CS. Thank you.
Thank you, Mr. Elder. What do I do here? We usually talk about safety and effectiveness, not effectiveness and safety. So we'll start with safety. It just rolls off the tongue better. First of all, we don't know who the anonymous people at FDA are who are hypothesizing side effects. Worsening a condition being treated. Well, everyone knows that every antidepressant doesn't work. That knocks that one out. Potential adverse effects from electrical stimulation in the brain. We've been in business 31 years with this technology, legally. There's no surprises coming up. The newer brain technologies, deep brain stimulation, TMS, VNS, these things are unknown long-term. Alpha-Stim is not. Potential risk of seizure, just take that one off the list. It hasn't happened in 31 years. Why are we still rehashing hypotheticals from the 1970s? Skin irritation, that occurs. As Dr. Earthman said, when you're treating a depression patient, they get a little bit of rash on their ear. It's treated with a cortisone cream, goes away the next day. So what? Headaches do occur, rarely, and they're self-limiting and mild. And blurred vision, cross that one off your list, it hasn't occurred. So let's get away from the hypotheticals, finally, that the FDA keeps rehashing in my 30-year career. And let's talk about truth. So now we'll talk about truth. First of all, in 1974, the FDA commissioned, as one of the public speakers said, commissioned the National Research Council in Washington here to review the safety of electrical stimulation to the brain, CES, and they determined that under 1 milliamp, that side effects are virtually non-existent. And there is no evidence to the contrary since 1974. The highest output of our devices is 0.6 milliamps, which is 60% of what the National Research Council considered to be safe. Now, in the last 5 years, 2007 to 2011, and I remind you again, we've been in business 31 years, we have sold 58,000 Alpha-Stims. And there were 15 reported side effects to us, mostly skin irritation, very strange one, black tongue, that turned out to be Pepto-Bismol. You live and learn. We also do a lot of surveys, post-marketing surveys of practitioners and patients, and we determined that, using a very conservative estimate, 8.25 million treatments have been conducted with Alpha-Stim over the last 5-year period. And we have 15 side effects. In addition, looking at every study that discussed side effects with Alpha-Stim, we did something that's very strange. We totaled the side effects, and we got 2.47, except they're not side effects. Of the 2,400 people in these studies who actually received stimulation, there were 59 reports. However, when you subtract the normals, like, oh, I feel the ear clips, or I feel a little dizzy, as you just saw on the video, that's normal for this. You're supposed to turn up the current until a slight vertigo, like rocking on a boat, is experienced, and then you turn that back. That's the 8th cranial nerve, which happens to be in the pathway of the stimulation. And so when you subtract out those, it's less than 1%. This compares favorably with TMS, and I know there was somebody who said that our TMS information is wrong. It is not wrong. It is taken from the FDA's presentation on TMS, which appeared that the FDA has stock in TMS, if you read that transcript. Anyway, they had more than half the people have a headache. We don't have anything like that. We use a very, very low current. So as far as serious side effect goes, in 31 years on the market, we have none. Absolutely zero. So I think it's past time we should be talking about fabricated, potential, theoretical, hypothetical, or fictional side effects. Let's talk about reality. If a drug's on the market 30 years, you don't start to speculate what it might do. And we deserve the same. So This compares favorably with TMS as well, which did have a few, although not too many, major side effects. All right, as far as effectiveness goes, we're only talking about CES. We do agree with FDA and Dr. Roche that there is no class effect from CES. The stimulation parameters are different. The FDA is talking about, because of that, you can't establish special controls. I don't even understand that. Does one antidepressant drug have to account for another one's? It doesn't make any sense. The fact is, our technology works. Another company needs to prove that their technology works. And when they say that substantial equivalent does not require clinical trials, that is not true in our case. We have the evidence to prove that our 510K, original 510K, took 22 months and required repeated evidence of efficacy to establish it as substantially equivalent to pre-amendment devices. They always made us prove that our technology works. All of our studies were independently conducted. We haven't funded any of the studies. We just hope for them. We do loan the devices for the research. And typically, well, not typically, dosage equals current inversely proportional to time, which means the higher the current, the quicker the treatment. So for research, we turn it way down so you can't feel it, and the normal 20-minute treatment is conducted in an hour. This is the flow chart of how all the RCTs I'm about to talk to you about were conducted. Basically, what we see here is that the device is locked in the parameters of a longer time and a low current. Subjects are randomized as per the protocol into the active or the sham group. And this is just as good as a sugar pill, a placebo pill in a drug study, because you don't feel anything. You see the device count down. It all looks the same. The only difference is the serial number, and that could only be determined by the key that we present to the usually the IRB in a sealed envelope. And some groups have a third usual care or wait in line control group, and some groups have a crossover arm of the study. So before, I don't know what happened to the top of this slide. Something happened here. But this is, it says up here where you can't read, but you have copies, EEG changes in 30 subjects treated with a single 20-minute treatment of Alpha-Stim CES. And basically this was done at the University of North Texas. Here's the nose, here's the ears. It goes from blue is less to red is more. And on traditional EEG groupings, we see less delta and more alpha, which means you're more alert and more relaxed. This is the effect that people report from a simple 20-minute treatment. There are 3 Alpha-Stim CS devices here. Okay. Here's what one looks like. You're welcome to play with it. You're welcome to put it on. It won't hurt you. Don't worry about it. It's perfectly safe. You become relaxed. Okay, so these are RCTs in anxiety. I'm going to go through these. Dr. Kim did a study on preoperative patients. Now, if any of you have ever had any surgery or seen anybody have any surgery, you've probably seen that's anxiety-provoking. And so he found significantly lower scores at the end of the, from before and after the treatment. At the P is less than 0.01. Level and at a large effect size. As a reminder, Cohen's D effect size is 0.2 is a small effect, 0.5 is a medium effect, and 0.8 is a large effect. He also found lowered blood pressure in his patients, an objective measurement. Dr. Strange did a study on chronically mentally ill partial hospitalization patients who are usually excluded from this type of research because of course, they all have anxiety issues, or they wouldn't be on this chart, but they also have major depressive disorder, bipolar disorder, schizoaffective disorder, and schizophrenia patients. And she found significant results of P equals 0.02 with with a small approaching medium effect size. Dr. Cork and Dr. Lickbrown did virtually the same protocols. 6-week study with 3-week arms, the first one being a randomly controlled trial and then a crossover to an open clinical trial. And they both were done on very severe pain patients who had anxiety, and they both had significant effects in the primary measures. Dr. Cork saw anxiety significantly improved at the level of P is less than 0.01, and Lick-Brown at P equals 0.02 at a medium effect size. Dr. Winnick had a study very similar to Dr. Kim, except not only was it preoperative, but it was intraoperative during dental procedures. And this had both the dentist and the patient look at the anxiety levels using two different scales each, before, in the middle of, and at the end of the procedures. And he found significant effects in all measures by both the dental observer and the patient. Dr. Voris saw psychiatric patients who had significant effects at the P equals 0.0001 level with a very large effect size, 1.60. That's more than a full standard deviation. And they also had similar results in objective measures. There was one failed RCT study in anxiety. However, this was state anxiety, the anxiety you're experiencing now, like when you're presenting to an expert panel for FDA, or when you're waiting in traffic, you're stuck in traffic, or when you're at the dentist. Or a preoperative patient would have this as well. State anxiety, anxiety I'm experiencing now as opposed to trait anxiety, personality trait chronic. So this anxiety was measured a week after the final treatment, which basically created a washout period. However, Dr. Mellon has published 6 studies through the University of Alabama and within the Alabama prison system, where it is used extensively. Now, confirming these RCTs are some very well-controlled open-label trials by experts in the field. The first one, Dr. Brzezinski at UCLA, runs the Anxiety Disorders Clinic at UCLA, and is one of the leading anxiety specialists in the world. In fact, he recently published an anxiety study in the Journal of the American Medical Association based on a $20 million government grant. And he also lectures at APA regularly. And so he did a study, and he determined that anxiety scores decreased significantly on HAM-A at the P equals 0.01 level and a very large effect size, 1.52. And that was on the HAM-A, and then the 4-dimensional anxiety and depression scale also had very significant results at P is less than 0.01 and D equals 0.75. This is his chart, which he was kind enough to loan me for this presentation, not my chart. This was presented at the American Psychiatric Association in 2009 and published in the Journal of Clinical Psychiatry. You can see the anxiety going down very clearly from this chart over the 6-week period, and Dr. Bystrzycki, who I failed to mention is also a full professor at Harvard, did compare this to results. He found the results similar to that found in clinical psychopharm trials. Dr. Overcash did a study for an HMO in Pennsylvania and found that ratings of anxiety were significantly reduced in his patient population at the P is less than 0.05 level, as well as on objective measures. So he used psychometrics and physiological measurements. So when we look at all of that data pooled, we can average a 0.9 effect size, which is large effect size from these studies, and a 0.79 effect size from the physiological measures, which you might round to 0.8, and that makes it large.